record. All right, boom. All right, here we go. Let's try this again. So Josiah, what did Griffith study again?
Griffith studied the relationship between bacteria and if the mice would die or live.
Good. And then bacteria, we refer to them as what?
Germs.
Germs. Okay, so now the question was, what are bacteria really? Okay, what are bacteria? Okay, and so I'm going to give you a list of things. Okay, let's start with atoms. Atoms form molecules. Molecules form what? Area, what do molecules form?
Cells.
No, not yet. We call them big molecules. Okay, last semester. What are big molecules? What's the fancy word for big molecules?
Macromolecules.
Exactly, that's right. So macromolecules. Okay, so that's something that we studied last time. And it was hard to really understand that because you weren't on campus to build these macromolecules. And then there's the macromolecules that we talked about. There's 4 types. Okay, there was the lipids, there was the carbohydrates, okay, the carbs, okay, there was the proteins, and then there were the nucleic acids. All right. Okay, so that's super important. All right, now, and then what we need to understand is that these guys right here, okay, come together. All right, so let's put them to a point here. They come together. All right, and they can form 3 different things. All right, number 1, they can form a virus. Okay, so these things can form viruses. These 3 things can form, okay, single cells called bacteria. All right, or they can form single cells that are called eukaryotic cells. And those are the cells that make up our body. Okay. And then from here, I don't know if you guys can see it here, but let me move it forward. The eukaryotic cells make plants, animals, and humans. Okay, so this is very important, like understanding this. So go ahead and take a couple minutes and take a screenshot of this or a little something so you guys have it. All right. So so really, where are we studying? What is Griffith studying in this chart? What is Griffith studying in this chart?
Bacteria.
That's right. He's studying the bacteria. So that's why we need to talk about bacteria. And so now, and I just told you the answer to my previous question, right? So this was what Griffith was studying, right? Griffith studies, okay, bacteria. And what are bacteria now? You should know the answer now. What are bacteria?
Germs.
Well, yes, they're germs, right? But really, there's 2 types of germs. A virus is a germ. We can consider that a germ. And then these single cells are also germs. So like bacteria are germs, right? So these are the 2 types of germs. But what's a bacteria really? What's bacteria? It's right here. A single cell, exactly. Bacteria is just cells. They're just single cells. Okay, and what are bacteria made out of? Work your way backwards. Work your way up.
All those 4 or those 5. Oh no, 4. All those 4 things.
Exactly, right? So these 4 things can build, okay, viruses, bacteria, or eukaryotic cells, right? And then over time, it took billions of years to do this. And then once the eukaryotic cells came into existence, then it took more, millions of years later for these cells to start forming things called plants, animals, and us, right? And so through evolution, that's how we got from a eukaryotic cell to these three things. But Griffith, he is studying these guys, bacteria, they're single cells, basically. Okay, so that's super important that you guys understand that. All right, so now knowing that, let's talk about these bacteria that he was studying, okay? And so bacteria really are just cells, okay? They're cells, all right? Now, these cells, all right, and actually, let me see here. These cells, they kind of look like, all cells basically have an outside, all right? This outside, we call it the cell membrane. All right. Now, does anybody know what the cell membrane is made out of? Which of these four things is the cell membrane made out of? Is it 1, 2, 3, or 4? Which of these four things make up the cell membrane?
Is it nucleic acids?
Good try, though. At least you tried. Ariel, what do you think?
Lipids.
Lipids, that's right. So the cell membrane, the outside of the cell, is made out of lipids. So what happened in the previous video, what I did is I just put a bunch of L's. Okay, like that. And then what I did is these L's represent those lipid macromolecules, all right, that are needed to build the cell membrane. Why is the cell membrane super important? Logic. Use your logic. Why is the cell membrane important?
It protects what's inside.
Exactly. It's the walls of the cell. Exactly. It separates the outside from the inside, right? We don't want things getting in. Like, why do you guys have walls at your house? How come you just don't have all the windows open, all the doors open?
So people won't break into your house.
Yeah, well, yeah, so they can't just come into your house easily, right? People can still break in, but not, it's harder. Now, so a cell has a cell membrane. So that's what these L's represent, the cell membrane made out of lipids. Okay. Now there's this other thing that cells need. Okay, they need carbs. Now, which carb did we talk about last semester that's super important?
Amino acids?
No, that's not a carb. That's an amino acid. What's the carb the carbohydrate that we talked about last semester is super important for energy?
Glucose.
Exactly, right? So all cells are gonna need glucose, right? So a cell is gonna need glucose, right? So in here, I'll put some G's in here. Now, do you guys remember what glucose is needed for?
ATP.
To make ATP, right? So there's these things in your cells, right? And I'll call that the mitochondria. All right, and then what these mitochondria do is they need the glucose, and then what they pop out is that ATP that you're talking about, and this is the energy, okay, that the cells need to continue to do everything that they need to do. So all cells need this glucose, right? So L stands for lipid, G stands for glucose. Okay, now what's the next thing that all cells need and makes every cell unique, makes cells unique? What do you guys think? In our cells, okay, it's actually inside a special organelle called the nucleus. So that's your hint. What's next? What's inside the cell that every cell has that usually is housed and protected by the nucleus? What do you think,
Yeah.
Leo?
Protein.
Nope. Close. It's one of the four.
Nucleic acids.
Exactly. But which nucleic acid?
Wait, DNA.
Exactly, right? So all cells, okay, have this DNA, right? And that's what we're going to study, okay, this semester. Okay, DNA. All right, now who's trying to come in here? Matthew, but thanks. Yeah. Oh, almost messed up my mind. All right, so here we go. All right, so yes, so they have this DNA. Every cell has DNA. Very good. All right, now this DNA, all right, is super important and makes the cell unique. So let's put the DNA in here inside a eukaryotic cell. It's in the nucleus. All right, so that's a little detail. Don't worry about it too much right now. So now, so far, okay, we have this right here. All right, so this nucleic acid called DNA, that's the DNA. And now finally we need one more thing that we need that we need inside every cell. What is it? Put a P. What is the P?
Protein.
Exactly. Right? So the P's are the proteins. Okay? So the cells that Griffith was studying, they're like all the other, like all cells, okay? They're just it's a culmination of, or a group of molecules that have come together to do specific jobs. So that's all a cell is. It's just a collection of molecules, right? And so the lipids build the outside of the cell, right? The inside has the DNA, has proteins, that has glucose. Okay. Now, so basically this is what Griffith was studying. He was studying two versions of bacteria. He called one the S strain, right? The S strain. All right. Why did he call it the S strain? Because when he looked at it under the microscope, they look smooth, right? So that was super important. Okay, so that's super important for you guys to understand right there, smooth. And now what did the S strain do to the mice?
It gave them I forgot what it's called
You got it. Start with
forgot.
it's the illness, right? It
Yeah.
caused the mice, it basically caused the mice to what?
Die.
Exactly. So the mice died when this cell went inside of them, because what happened was this cell gave them pneumonia. Okay, gave the mice pneumonia. All right. Now, there was another cell there was another type of cell another type of bacteria that he studied, right? So it's going to look, it's going to have the same thing. It's going to have a cell membrane made out of lipids. There it is. Right? Lipids. All right. Now, okay, now this other cell, he called it the R strain. Okay, because under the microscope, it looked kind of like, it looked rough. That's what he said, right? And if you look at the text, you can see that. Now, the cell is made out of lipids. Do you think this cell needed glucose? What do you guys think?
Yes.
Yeah, it's gonna need some form of energies, right? So it's gonna need some molecules for energy, right? So it's gonna need that, right? Okay, now what made the S strain and the R strain different? What do you guys think? What's gonna make them different? Okay, how come? What makes them different, guys? Exactly, right? What makes Ariel and Jeremiah different? What makes Ariel and Leo different?
Their DNA.
Exactly. So this is the same concept with cells, right? These guys had different DNA. They had different codes. And then when these codes, okay, what do these codes help you make?
Proteins.
Exactly, right? So let me go ahead and draw the proteins. Here we go. I'm gonna put them a different color, the P is different color, because they came from a different piece of DNA. So that's what's happening inside your cells, right? So where's my black marker? Okay, do I have a black one? Let's see, this is a gray one, that'll work. So what happens is DNA has codes that help you build a protein. DNA has codes that help it build a protein. And so what ended up happening, and this is what I want you guys to leave the class with today, is that when this bacterial cell went into the mice lived. Why did they live? Because those proteins that this cell made didn't cause pneumonia in the mice. So that's why they never got pneumonia. They didn't get sick. This guy, however, has DNA that makes proteins that made the mice sick. And so therefore, the mice died. So it's very important that you guys understand this. All right. Now, why is it important that you guys understand this concept of the experiment? Because what ended up happening? What was the end result of the experiment, of the Griffith experiment?
Wait, like the last part?
Yes. Remember this? Remember this from yesterday?
Yeah.
What was the end result?
Oh, the mice died.
No. What was the part of the experiment that was like super important to understand and that it was kind of groundbreaking? What did the harmless bacteria download?
DNA.
The DNA from who?
The hold on, I have it on my notes.
It's right here in front of you.
From the fire?
No, from the fire, no.
I meant the. To be honest, I don't know. I'm kind of confused now.
Exactly, right? So here's the experiment, basically, right? The smooth bacteria caused the mouse to die. The rough bacteria caused the mouse to live. Okay. When he took the smooth bacteria, heated it up, that killed the bacteria. And therefore, when this stuff got injected, the mice lived. Then what happened is he took the same juice, added the harmless bacteria. Now you have two things that don't do anything to the mice, don't cause the mice to be sick. But when this stuff got injected into the mice died. And so Griffith hypothesized that, okay, remember what cells are, right? Remember, this is what cells are. They're basically a collection of molecules, right? They're basically a collection of molecules. So what ended up happening was somehow this DNA, okay, right here, got downloaded by the R strain. Okay, it got downloaded, okay, by the R strain. And the reason why I use downloaded is because you guys understand what download means. That means that you guys are putting something into your computer, right? You're taking something in. So what ended up happening is in this beaker right here, the cell, because it was dead, basically think of it like the lipid bilayer, okay, basically got broken up. And so this stuff was able to come out. And somehow this DNA got downloaded in, like this R strain took it in. And if you can imagine that this green DNA now is in here, like this. Okay. And now that caused this R strain to build proteins that before it didn't have to build. It didn't have codes to build. And now these new proteins, because it has these new codes, transform this cell into an S strain. That's what happened. And so when this juice got injected into the mice, that's why the mice died, because the DNA from this guy got downloaded into this guy, turned this guy into a smooth bacteria that was making the proteins that caused the mice to die. So hopefully that makes sense. And when you play this back, hopefully you can follow that through. But it's very important that you understand all these steps. But if you don't understand this part right here, okay, what a cell's made out of, then you're not gonna understand, you know, the download of the DNA and how that DNA now is in this cell that didn't have it. And now because it has these new codes, it has these new proteins, right? Remember, we talked about proteins like apps, right? Think of a protein like an app for the cell. And these apps, these proteins allow the cell to do things. In this case, it allowed the bacteria to make a protein that was toxic to the mice and caused the mice, right, to no longer like not get sick, but it caused the mice to die because of these new codes that allow it to make these new proteins. So hopefully this makes sense. If it doesn't, then you know, stick around after class and we can go ahead and talk about this. Do you guys have any questions so far? Questions, concerns on this? Questions, concerns? All righty then.